Welcome to this explainer. Okay, today we are diving into a really fascinating topic called glial timing architecture, and we're basing this on an incredible manuscript by Pierre-Auguste Jeanty. Now, for decades, the dominant model of the brain basically pictured flashing neurons doing all the heavy lifting. We kind of just thought of them as the solitary decision makers of human cognition, right? But as we're going to see, systems neuroscience is actually revealing a totally hidden, incredibly complex timing infrastructure right underneath that activity. So instead of a solo act, our cognition is much more like an intricate symphony. Let's pull back the curtain on the hidden conductors of the brain symphony and explore how these non-neuronal systems are the ones actually orchestrating our thoughts.
To kick things off, we really have to ask a fundamental question that challenges the classical view of how our minds work. What if the star of the show isn't actually the neuron? I know, for so long we've been taught that neurons are the absolute center of the nervous system's universe. But there's a massive paradigm shift currently underway. We're discovering that higher order cognition isn't just about whether a neuron fires or doesn't fire. It's about something way more nuanced. It fundamentally relies on the precise coordinated timing generated just beneath the surface of that electrical spike.
So let's jump into section 1, Beyond the Neuron, where we are completely rethinking the brain's blueprint.
Historically, classical neuroscience heavily emphasized neurons as the principal computational units. The traditional belief was super straightforward. Neurons fire, they form networks, and boom, that creates a thought. End of story. But the emerging field of systems neuroscience is showing us that higher-order cognition isn't nearly that simple. It actually depends on meticulous, coordinated timing regulation that's distributed across both neuronal and non-neuronal biological systems. Just think about it for a second. To generate a complex memory or smoothly execute a brand new motor skill, signals from vastly different physical locations in your brain have to converge at a specific destination at the exact same time. To pull that off, your brain requires non-neuronal cells to meticulously coordinate the exact arrival time of those neuronal signals. It completely shifts our understanding from a purely electrical model to this deeply integrated bioelectrical timing system.
Which brings us nicely to section 2. Oligodendrocytes and adaptive myelin, the actual timing infrastructure of your brain. To really grasp this infrastructure, we need to immediately bust a major scientific myth. For the longest time, science textbooks basically compared myelin to the passive rubber coating wrapped around a copper electrical wire. It was viewed as just this inert insulation, simply there to keep electricity from leaking out into the surrounding tissue. Well, under this new architectural framework, myelin is absolutely not just passive insulation. Treating it as some static anatomical feature completely misunderstands its biological brilliance and its super active role in human learning. Instead, you should think of myelin as an active master conductor. It dynamically regulates axonal insulation and it meticulously controls nodal spacing. Those are the critical gaps, known as the nodes of Ranvier, where the electrical signals actually jump. By physically altering its own structure, changing the thickness of its wrap or adjusting the width of those nodal gaps, adaptive myelination actively adjusts the propagation speed of action potentials. Basically, this infrastructure can intentionally speed up or slow down electrical signals originating from totally different parts of the brain. If one signal has to travel further than another, the myelin tweaks the speed of both so they arrive at their destination at the exact same millisecond. This isn't just static insulation, it's a wildly dynamic timing infrastructure keeping the whole network perfectly in sync. Okay, moving on to section 3, OPCs as timing regulators and dynamic neurodynamic control. So how does this crazy regulation actually happen in real time? Well, oligodendrocyte progenitor cells, or OPCs, function as the active managers on the ground. First, they receive direct synaptic input from the neurons themselves. They're literally woven right into the network, actively listening in on the electrical chatter. Then, upon receiving this input, the OPCs generate intracellular calcium signaling. Think of this as an internal processing alert that translates the neuron's electrical activity into a chemical signal inside the glial cell. Finally, they use this process information to physically regulate adaptive myelination. They're responding directly to what the network is doing, processing what the system needs, and then dynamically tweaking that physical timing infrastructure. They are the active regulators constantly fine-tuning our neural systems. Next up is section 4, astrocytes. These are the network coordinators managing distributed synchronization. Now, this vast system of constantly speeding up and slowing down signals requires massive, overarching coordination, right? Otherwise, it would just devolve into total chaos. And that brings us to astrocytes. Astrocytes are specialized glial cells that manage synchronization, stability, and neurodynamic coordination across all these distributed systems. If OBCs and myelin are the ones adjusting the individual speed of the players in our neural orchestra, you can think of astrocytes as the ones managing the acoustics of the entire concert hall so the whole system stays stable. They oversee the broader biological landscape to make sure that local timing tweaks don't disrupt the global harmony of the brain. To maintain that stability, astrocytes literally do all the heavy lifting. They regulate the extracellular ion balance, which ensures the exact chemical environment needed for neurons to fire optimally. Without that, action potentials would totally misfire. They also actively manage neurotransmitter clearance, sweeping away residual chemicals from synapses so you don't get noise and interference between communication cycles. Plus, they provide the underlying metabolic support, basically delivering the energetic fuel needed to power the entire network. They even regulate oscillatory environments to keep large-scale brainwaves synchronized, and they facilitate broad communication across the entire glial network. They're doing so much crucial work just so cognition can occur seamlessly. And this leads us perfectly into Section 5, the ECPJ Systems Framework, which is the grand unification of everything we've talked about. So the ECPJ Systems Framework is this overarching systems-level model that beautifully ties all these elements together. It integrates classical electrophysiology with adaptive conduction, glial regulation, and distributed neural coordination. Janti's framework really provides a grand unification for systems neuroscience. It forces us to stop looking at the brain as just a simple network of electrical wires, and instead see it as a deeply integrated biological machine. In this model, electrical signals absolutely do not act alone. Their timing, their speed, and their ultimate cognitive impact are perpetually sculpted by these non-neuronal glial cells operating quietly in the background. Now, you might be sitting there wondering, okay, this is neat, but how does all of this microscopic cellular biology apply to my everyday life? Why does this specific glial architecture matter for the exact thoughts you are having, the sentences you're processing, and the memories you're recalling at this very second? Well, it matters because, as the research so clearly points out, cognition likely depends on adaptive timing coordination distributed across both neuronal and non-neuronal systems. Every single thought you have, every memory you pull up, every new skill you try to learn, it doesn't just rely on a lone neuron firing in a void. It relies on a perfectly synchronized, highly adaptive dance between those neurons and your hidden glial network. Your oligodendrocytes, your OPCs, and your astrocytes are constantly shaping the physical architecture of your mind to make sure your thoughts arrive with absolute pinpoint precision. And honestly, if our very consciousness relies on this precise biological timing, just imagine what new scientific and medical frontiers we could cross if we learned to fully master the hidden conductors of that timing. It's a pretty remarkable concept to leave you with, and definitely an exciting direction for the future of neuroscience. Thanks for exploring this with us.